Hello, everyone. Once again, this is Mike Grossman from BioPharm Systems. I did want to thank everybody for taking the time for joining this teleconference and web conference. A little bit of background. I'm the VP at BioPharm, responsible for the clinical warehousing and analytics practice. In this web conference today, we'll be talking about using Oracle's Life Sciences Hub and looking at something called the dynamic cross-study analysis. Some logistics before we get started. At several points during the presentation, I will be breaking from one agenda point to the next. But as I go through the presentation, please feel free to just type your messages in the chat window, and I'll take a break between each section of the agenda and try and answer as many questions as possible. You certainly can wait until the end, but some of the topics may be out of context if you wait till the end of the discussion. So please feel free to use the chat function and ask questions. I may not be able to get to all of the questions, but hopefully I will. Also, this presentation, along with the audio portion, will be recorded and will be available for you to listen to later. Also, if you have colleagues that you'd like to share it with at some later date, they also can register after the fact and listen to and watch the webinar as it moves forward. So with that, I thank you once again, and I will jump straight into the presentation. So I'm now moving on to slide 2. So for those of you that are watching the slides online, you can get an idea of how fast the screen refreshes. Today, our agenda, we are going to, within an hour, fit the entire agenda. Really, I'm going to talk a little bit about what do we mean by dynamic analytics, what are some approaches that can be used for dynamic analytics, how do you prepare data for it, select data, build models, how does LSH fit into this overall framework and approach, and then just a general session at the end for Q&A if I am not able to answer all the questions as we go through the presentation. So the first section that we're going to be reviewing is just what is dynamic analytics. What do we mean by that? So when I'm talking about dynamic analytics, I'm talking not about on a clinical trial or a clinical submission, the formal gathering of the subject data and preparing that for submission for approval to regulatory authorities. That's not what I'm talking about with dynamic analytics. What I am talking about is the ability to bring together groups of data, patient subpopulations based on a specific criteria that's independent of the fact that they were conducted in a specific study or they were part of a specific submission, although that certainly can be one of the characteristics. And the ability to combine that data and analyze that data for a business purpose beyond submission. So I just have some examples here that I've listed just to kind of give some context as to what we mean by dynamic analytics. So the first main section here talks about study and program feasibility. So as you're planning out your portfolios, your programs, and your studies, the types of things that you would like to be able to do with a high level of accuracy is to perhaps predict what is your chance of enrollment success based on patient populations that exist within certain regions, certain characteristics. If you know the prerequisites for the subjects, what are the inclusion-exclusion criteria that you are trying to put together? Will you be able to recruit that patient population over time? And what will be the cost of that recruitment? So through advanced analytics and actually looking at previous trials and information embedded within previous trials for certain disease types, for certain criteria, you should be able, using analytical methods, if you could get that data at your fingertips, you should be able, in theory, to analyze different scenarios, different conditions, and actually what's it going to cost for recruitment? Is it possible to get the recruitment you're looking for? Where is that recruitment going to come from? Another example is predicting risk factors. So you may look across historically and for current trials, compounds of a particular type. And you may want to compare that with maybe some complications involved with other factors, such as demographic factors, other diseases, other concomitant medications. And you don't want to restrict it just to the studies necessarily that are occurring on the compound that you're planning on putting trials together on, but you may actually want to compare that with outcomes data, and you may want to compare that with other trials of drugs of a similar class or purchase databases. Now, this has been a goal in the life sciences industry for a fairly significant time, and many companies have been successful in producing one-off projects that prove this is theoretically possible. But the industry overall has had a difficult time in systematizing these kind of questions without making each question a special purpose project. So what are we actually talking about? Well, we're talking about the whole process of looking at your legacy study data, outcomes data, ongoing trials, and preparing that data into getting it into some sort of shape that you can actually analyze it. Also, we're looking at selecting subject or patient subpopulations based on a set of criteria. So you need to look across all of your clinical data, set some criteria, and be able to essentially mine out the subjects that you're interested in looking at for a much more extensive analysis. And then once you have selected that subset of patients, you then want to look at all the data that you've collected for those patients over time. So not just based on the factors that you've subset by, but the full history, longitudinal data of that. Then you want to apply, build models, and train and confirm your models against that data for the particular question and hypothesis that you're trying to ask. Once you've done that, you may want to apply those same methods against different subpopulations, or you may want to use multiple methods and answer multiple questions against specific subpopulations. And so that's an iterative process that's continuously being reevaluated. So what we're really going to talk about today is there a way of doing that? And the key question is, most companies are capable of doing this with a brute force method on a one-by-one case approach. If they have a very specific hypothesis, they can dig back into their legacy data, find it, write programs against it, and answer a specific question. But it's very, very labor intensive. So what I want to focus on today is there a way to establish a systematic approach to this, so that as you set up every single trial and as you bring it live and bring it to production, it suddenly is available to answer these types of questions along with the pool of historical data that's already been set up for this type of approach. So can it become the production mainstream that even if I don't know all the hypotheses of the questions that I'm going to ask, I have an environment that's ready to answer these questions in a rapid and, I wouldn't say low-cost manner, but a reasonable cost manner in a very repeatable and reportable way. Okay, so I've now moved on to the next. I'm moving into the next section. I've gone beyond the overview, and now moving into let's drill a little bit deeper and talk about this approach with dynamic analytics. At this point in time, I don't have any specific questions. You should feel free to ask questions, and there has been time allotted for that. And as I go through each one of the sections, I will go through and answer questions. And in order to ask the questions, you just can use the chat utility, which should be in your control panel for the presentation. So the next is, how do we approach dynamic analytics? So we've taken a hard look at BioPharm, at what this entails, and seen that there really is a common pattern to how you approach these kinds of problems, potentially. So really there are 4 key stages that we've identified. And this is not something that's specific to BioPharm. This is something that's fairly well understood in the industry in a general sense. So the first is you have to prepare your data. if all your data is scattered all over the world in different shapes, different sizes, different systems, the amount of effort it takes to pull together the data that you're interested in is so high that it becomes cost prohibitive. So we're going to talk a little bit about data preparation. Once you prepare the data, you do a preliminary exploration. And that preliminary exploration basically is to find a subpopulation that you're interested in to begin to train your model. So once you've found that initial subpopulation, and you've done the exploration through the data to find that subpopulation, you then would create a longitudinal view of those subjects, and you could start building your analytical methods against that subpopulation. Once you have an initial analytical method that you're happy with, you then would want to redistribute that. You might want to tweak that analytical method and create a second one and a third one to look at variations on those analytical methods. You may want to take those analytical methods and you may want to run them against different subpopulations to help verify if the assumptions you made in building the initial methods were in fact correct. Once as an individual you've gone through that iteration, you then may want to record the results, save those methods, save those subpopulations, and then distribute that out to other teams within your company and other colleagues so that they can learn from what's there, and that can be stored and be shared as a company-wide asset rather than as something that is sitting on a Unix workstation, perhaps in an individual's office. The next slide kind of starts to show some data flow representation of that rather than a process flow. So what you may have, and this is just a possibility, is you may have EDC data and other data coming in. You may have historic data coming in maybe via files. And we are recommending that that go through a data standardization process. And I'm going to talk about each one of these steps in detail as we go through the talk. Once you have, at a study level, you've prepared that data into a conformed structure, you would then want to pool up a subset of that data so that it can be interactively explored, and that's step 2. So you can pick your subpopulations that you're going to use your methods against based on specific criteria. And we're going to take a little time to go through that and ways of handling that. Once you've selected in step 2 your subpopulation, you then essentially want to go back to that subpopulation and go into all the studies for that subpopulation and pull all that data out, the full history, that longitudinal data of those patients, and then start building your analytics methods. You want to save those results and then reuse various combinations of subpopulations against the library of methods that you build up over time. And the concept really, one more slide, is to build that on top of a more general architecture that not only covers dynamic analytics, but it's an IT reference architecture for clinical. It's a reference architecture for clinical IT that supports both the day-to-day operations, clinical trials management, data management, biostatistics, project management, etc. So there's a general architecture that you'd like to fit it in so that this is not an activity that happens off to the side, but it just happens it can happen in the mainstream of running trials. Now, at BioPharm and in other places, there is a pattern that is appearing in the industry that basically is a 3-pillar pattern that says take your transactional systems, stage that data up as is, so take your EDC raw data, take your outcomes data, take your adverse events data, stage it up as it exists in the individual transactional systems where it's collected. Bring that data together in an operational area so that, in fact, you can normalize the data, standardize the data, and integrate the data. And then once you have done that, you can then have special purpose data marts, which is really what we're talking about today, of warehouses and mining and dynamic analytics. And there's a whole tool set around that architecture. And this is not an architecture that's built overnight. It's done project by project, piece by piece, each step having a return on investment. And we're really focusing on the project of dynamic analytics. I have a question here from one participant asking about, are there pre-built metrics or methods that can make prediction on a new study? And this particular presentation is not discussing a library of pre-built metrics that can be used for specific business purposes. The idea of this presentation is really to focus on how do I build up that library of metrics over time, and metrics and methods over time, and systematize that so that I don't have to go back to raw data and collect the data back every single time. And I have a full, traceable, production-ready way of building those methods. There are a number of projects that are actually out there that are actually looking at a standard set of methods. One such project is the OMOP project, or Observational Medical Outcomes project. I'm not sure I got the abbreviation, it's OMOP, that basically is looking at outcomes data and actually building out a set of standard methods that can be used for various types of predictive analytics. This specific talk isn't going to give you those specific methods. This specific talk is really how do I build an infrastructure that lets you build that library of methods over time so you don't lose that knowledge as you go from project to project. Okay. So the first step really is data preparation. How do I prepare the data? So what's involved in preparing the data? Gathering the data. So EDC, historic data sets, SAS data sets, other internal sources, even external sources. You have to get those in to maybe not the same physical location, but a common location, so that every time you want to do some dynamic analytics, it doesn't become a hunting expedition to find out where that data is coming from. The next step is that you need to conform the data. So the goal of conforming the data is that the data has a common shape and that you have a better understanding of the data without going all the way back to source. And then you need to take that data and you have to prepare it in such a way that you can move on to subsequent steps that allow you to do the cross-study analysis. So I'm going to drill into a bit more detail here. And specifically on the conforming step. So staging the data up from the individual source systems is something I'm not going to cover in detail. That's kind of a separate talk about how to stage and conform data that would require its own discussion. Many companies are using Life Sciences Hub to stage the data and then conform it for many purposes. And many companies are doing this and they don't use Life Sciences Data Hub. They use other products. There are other products on the market. And some companies are doing that as a bespoke approach. So generally the focus is on EDC data and other study data because you're getting with each clinical trial, you're getting new data and new data structures in. In theory, any standard conformed target structure would work. Each one of them has pros and cons. So you may have a company data standard that you've been working with for years, and that's fine. Most new projects or companies that we're working with now, actually when they look at a standard structure that they're moving the trial level data to, they take a CDISC SDTM 3. Specific version 3.1.2 or whatever version they're working with, and then they add to it. So what's typically called SDTM+. There's a number of reasons you would want to choose SDTM+, but it's not perfect. And I think for people who have tried this, they know it's not perfect. One of the key advantages of SDTM+ is that you don't have to necessarily start with a white sheet of paper and get everyone within your analysis community to agree what it's going to be structured like. So if you start with SDTM and then you start adding the bits that SDTM doesn't cover to what you need, it's usually a shorter conversation. So agreeing on a standard instead of taking months and potentially years actually can take weeks. Also, if you use SDTM+, the chances that you could use the data for other things, such as preparing your submission, is greater. And as you have personnel changes in your organization, many people can be hired that also are very familiar with the SDTM structure, so you don't have the same training and learning curve that you have with some other standards. So there are some significant advantages to SDTM+, but there's no reason specifically why it has to be SDTM. The other thing to note is this conforming step does not exist purely for the existence of being able to do dynamic analytics. These same conformed data sets or same conformed structures can be used to assist in data cleaning. They can be used for your formal statistical analysis that you're using for submissions. If you have standards listings and reportings for operational purposes or medical review, all of those can be excellent starting points if you have a conformed set of your clinical trials data. What we're going to be talking about today is how this conformed data is being used for dynamic analytics. But it's not that you would conform it specifically just for that one purpose. And that's very important because the effort required to conform the data, which really means that you take some time up front, as soon as you're setting up your EDC trials or other trials, you actually go through the effort of conforming the data. That cost, actually benefit calculation, really needs this conforming data to be used for multiple purposes and not just for a single purpose. Another experience point that we have on preparing the data and conforming it is very often you want to get the meaning of the data to be identical in all cases. Now that's challenging and may not be practical, so I'll give a specific example. You may decide that for blood pressure, the way you would measure it is you're going to use a vital signs findings structure based on SDTM, and you want to model it as systolic blood pressure, diastolic blood pressure, and the position that the blood pressure was taken. But in fact, that's probably insufficient to document all kinds of blood pressure, because there are many ways of taking blood pressure, and those three fields themselves are not sufficient to cover every possibility. But you have to be a pragmatist, so I recommend initially, when you agree on the standard, that at least you get the shape to conform, recognizing that for 100% of data, there's still some risk in pooling that data, that the meaning might not be identical as the shape. You would focus on getting the meaning the same in the following areas. First of all, get it in the same shape. If it's the same basic medical item that you're measuring, try and unify your terminologies. So this really has to do with using MEDRA, WHO drugs, code lists, and units. If you can get that meaning to be the same in your first pass, as that standard changes over time, you can get better and better at making sure you have a unified meaning for all your medical terms so that it really becomes very, very poolable in a medical sense. If you try and get everything to mean exactly the same thing at the beginning of your project, it may take you so long to get agreement that it may not give you your full return on investment. So those are just some pragmatic approaches when preparing the data and getting it ready. And at this point, you basically would have separate containers, schemas, directories, whatever technology you're going to be using, and that would have essentially identical structures in them with data from each study. In the case of using the Oracle Life Sciences Data Hub, these would correlate to work areas. So you'd have a conforming work area for each study, and in that conformed work area, you would have the data for that study, whether it be a combination of lab data, central lab data, EDC data, specialty labs, PK data, randomization, the full set. That is your conformed data that has a longitudinal view of the patient. So again, no new questions. Please feel free to type them as you need. So now I've got these conformed data marts, one for each study. Potentially we also could have conformed data marts with outcomes data that are not study specific. That's fine. The next step is data selection. So what do we really mean by data selection? Well, you want to look at a subset of the data based on study and subject characteristics so that you have a test subset of subjects cross-study that you can actually build out your methods and hypotheses on. Additionally, you may want to identify for your hypotheses the specific grouping, once you've picked your subject subset, of variables that you want to pull out. Or alternatively, you could just pull out every single variable across all of those patients. So I'm going to drill into quite a bit more detail on this. Really, it's about this data selection. This is something that's really been a struggle that many companies have had difficulty systematizing. So really, the user experience that you want in creating these subsets is not to write complex programs and submit them and have it troll through all the data and come back. In the ideal world, what you want to do is you literally want an interactive session. You want to sit at a dashboard on your computer, and you want to just pick criteria about your subjects and pick criteria about your studies and have it interactively in front of your eyes build these subsets. So there is a way of doing that, but it does require some planning and design. First of all, the subset building is not going to be based on every single characteristic of every single study. You pick a group of characteristics that you're interested in on your study and a group of characteristics, subject domains essentially, that you want to prepare your subsets from. Now, because this interactive experience is really going to expand all the data across all your studies, and you're not going to be restricted to one or two studies, the actual technical performance of that interactive experience needs to be taken into account. So you can imagine if I had a pooled set of all my lab data, for example, that would be hundreds of millions of rows if you were a large company and had a large set of data. Now, interactively working through hundreds of millions of rows doesn't really work if you just leave things in an SDTM-like structure. It was never designed for that. Computer systems really, without making major investments, are not really designed for that either. So there is a technique in building out warehouses called dimensional modeling that does allow you to actually slice and dice data of extremely high volumes like that. And the way this dimensional modeling works is you pick a topic or a domain or a fact, group them together, and think of the ways that you want the end user to slice and dice them. Now, in initial implementation, you start with a smaller set. Once you get experience with that, you may want to add some sets. So the list I have here is just a candidate list to begin a discussion. But the way that you want to prepare your patient subsets, you clearly need study characteristics. You want to say things like, give me all studies within a particular therapeutic area, or that were a particular compound that has an active ingredient, or used a particular device. They were submitted within, they existed within a certain date range, those types of things. So you have to know about the study, what phase it was, etc. Very often, you want to identify what was the inclusion-exclusion criteria used for a particular trial, and what subjects passed or failed that specific inclusion criteria. You also would want to look at the demographics domain. I want males, I want females, particular race, etc., particular age groups. You would want to look, as a minimum, at exposure and conmeds, and I do combine those two domains on purpose, because when you're looking cross-study, the actual difference between the exposure data set, where you're talking about the active research item, versus something like a conmeds, which were other drugs that were taken, is not necessarily relevant. You really want to look at those together. The fact that they were exposed to a particular compound, whether it was concomitant or whether it was the primary one, is not critical in this analysis most of the time. And very similarly, the difference between adverse events and diagnoses for this purposes is not critical. So you'd want to actually pool those together and treat them as one thing so you could cut them the same way and combine them. Because an adverse event, or primary adverse event in one case and primary indication in another case and a diagnosis in another case really might just still put you into a position where you want to include that subject in your longitudinal analysis. So these are the topics. Let's talk a little bit about how that data needs to be structured. So these are just examples, just to help illustrate the point and explain how this works and why it performs so well. There's something here that's used in warehouse terminology called star schemas, or just stars. And for each one of those topics, what you do is you set a table in the center that basically points to all the different characteristics. So in the center basically are just numbers. And if you have a particular primary indication, you would tie that to what's called a dimension in MEDRA. You'd have the study name as another dimension. You'd have what phase the study is as another dimension. What program, what subpopulation if appropriate. What regions was that study run on? And what compound or device was actually used in that particular study. So if you wanted to say, I want to see all phase 3 studies, in that study fact, that list of rows that would have every single study that you use in your entire inventory, as soon as you say phase 3, it restricts that study fact to just the phase 3 studies. Now that happens very, very quickly because when you look at the choice list for phases, it doesn't scan through the full list of studies. You've already, in the phase bubble that you see there, got your 4 or 5 or 6 unique phases that you have, and you pick from that list, and it very rapidly narrows down the list of studies. And I'm going to go through a couple of other stars and talk about why this works across stars and how you can very rapidly go through millions of records to come up with your subpopulation. This is an example of a demography fact. Again, the study fact really, the level of granularity is per study. The demography fact, typically you would correlate that to 1 record per subject. You'd have information about what study they were in, perhaps you would also tie that to the site or region that that subject was associated with, the gender, their subject ID obviously, what race, maybe the age in years if you want to do range, age range subsetting. So the idea here is I could very rapidly say, give me all males, and it will immediately reduce the list of subjects in the DM fact to just the males. I've got a few more. I'm not going to read each one of them out loud, but just to quickly kind of go through, it's how can I slice and dice. So the idea is I will pool together all my ConMeds and all my exposure records, and I'd pull out maybe what study they were on, what subject they were, maybe what was the dose form, what was the drug that they were exposed to, maybe some start dates, end dates. That's kind of optional whether you want to do date analysis, root of admin. So that very, very rapidly in this star, I could easily just say, give me, anybody that took an analgesic, and immediately I would get the list of studies and subjects. But I think the key here isn't within a single star can I get that answer, because in theory you could come up with an indexed optimal way of doing that. The power really comes as we move into the more complex cases. This is just an example of an AE star. Again, severity, these seriousness, MEDRA, etc. The real power comes when you do something called shared dimensions. And almost all of these stars have shared dimensions on study and subject. So the goal really is, if I wanted all analgesics on the AE fact, that is tied back to the MEDRA preferred term hierarchy dimension. It will very rapidly reduce that list to just the AEs that were and diagnoses that were associated with analgesics. That intersect gives me that unique set of studies and subjects. But if I'm using that same dimension, the same study IDs and same subject IDs in my dimension fact, in my DM fact, my demography fact, I can now subset it by race. And I'll get that list very, very rapidly. And this can be across millions and millions of records. So now the end user experience is they can start combining things between diagnoses and AEs, demography, study characteristics, etc., in an interactive session. This is just a brief screenshot. I know it's difficult to see. It's a live database, but it's not exactly a perfect implementation. Also, it's clipped off on the side. But the idea is that an end user, and this is using a tool called Oracle Business Intelligence, OBIEE, that's layered on top of LSH, where basically these stars sit underneath, and an end user can just go and pick the variables they're interested in, and then they can just start applying filters to those variables, and the performance is extremely fast. So you basically pick, you assign your filters, you say display results, and you'll get a very rapid list of studies, subjects, as well as all the extended attributes that you filtered on. Now, in order to prepare these stars, we use, in the case of LSH, automation to go into the individual conformed studies and automatically add those studies to these stars, to this dimensional model. So there's not a human intervention that's required in order to prepare the stars as new studies come online. Once that subsetting is done, you then want to export what your subset was. You probably want to save it and then export it. And essentially take that subset and push a button and have it automatically go back into your longitudinal conformed data for that subset and prepare you a pooled set of longitudinal data with just those subsets. So what this is actually showing is the first rectangle on top involves the preparation stage. So this is something that happens within Life Sciences Data Hub, something that's done on a study-specific manner, where you stage the data from EDC and then you conform the data after it goes through a validation life cycle to some sort of standard. And for today's discussion, let's just say it's a version of SDTM. So you've got SDTM data, or some version of SDTM data, in production, ready to go. That's part of study setup. You then run a program that basically loops through those production conformed data and then adds that data to the pooled stars. And those exist in the second box, second set of boxes. In the second set of boxes, below the first set, you have the stars. You do this interactive subsetting. You save your subset, upload the results of that, kick off another program, and it will automatically go back into the conforming work areas of several studies, as many studies as you want, and pull out those patients' longitudinal data across all of their patient records and prepare a data mart that's specifically focused on that subject subset. So these are pieces of automation that we have in place. Okay. People that are not asking questions, please feel free to do so. Let's continue. So now we've got our subject subsets. Oh, look, somebody did that. That's fantastic. Okay. So for companies who are conforming data sets, are they using ADAM data sets as part of this? You certainly can. There are a number of variables in the ADAM data sets that are essentially imputed variables that you may want to use for two purposes. One is you may want to bring them into your stars for the purposes of subsetting. That's probably less likely. But certainly you may want to carry those extra imputations that you wouldn't have in pure SDTM into the longitudinal analysis, so that your methods that are actually looking to answer these questions could actually take some of those derived and imputed variables that you have in the ADAM data sets and actually take advantage of them. We don't actually have a specific use case in production where that's happening right now, but there are ongoing discussions about using ADAM as your standard rather than SDTM. As you might imagine, using ADAM means that you have to standardize all those imputations that generate the ADAM data sets, which can be a tricky business. So thanks for that question. So let's move on to the other steps. I see we have about 15 minutes, so I'm just going to move ahead, but the timing's about right. So the next thing is, okay, and let's build the models. Right? So you had a hypothesis, you've got the longitudinal data, you've got your subject subset, you actually have to build the models. And as I said earlier, this talk is not on how you build those models. Typically use a tool like SAS or R or something like that, and you'll build up these as analytical methods, and you'll run them against the subject subsets. And then we basically will take those methods that you're writing, and you'll iteratively run them against the subsets that you prepared in step 2. But once you get it, you think it's working against that subset, you probably have to try different subsets. So it becomes a very iterative process. And if it takes a long time to prepare those subsets and you have to go dig for more data and it wasn't an interactive experience, it could be very painful. So the concept here is it always is going to take time to build these methods. Either you've got them in a library somewhere because you got them from somewhere else, or you've developed them yourself and you want to share those methods. But we have to be able to quickly create subsets based on different criteria, and that's why we recommend that's a dimensional model that's an interactive experience. Now, once you've got a method that you think is usable, keeping it to yourself is not very useful. Because once you have a method, if you can parameterize it, work against different subsets, so perhaps you're looking at a method that actually looks for specific body systems or something like that. If you can pass the body system in as a parameter, that method becomes more general purpose. You'd like to store the knowledge that's in that method, as well as a record of what subsets you ran those methods against, so that you can actually save it, keep it under version control, potentially do some level of validation, and actually share that with the wider community. And the idea would be that companies can slowly build up libraries of these methods, and that you don't end up having people doing dynamic analytics, rebuilding methods that actually were built 2-3 years before, but people were just unaware that that method was available. So that's the goal. In Life Sciences Hub, the way you would do that, programs, whether they're SAS programs or R programs or whatever, are stored in a library. And then you apply those programs, those methods, against the subset. Now the subsets are basically, the longitudinal subsets are basically the same shape. It's idealistic to think they'll be exactly the same shape because your conforming standards, while they may be a standard, are still slowly evolving over time. But your chance of having a method run against different subsets over time is much, much higher if you start from a conformed and controlled standard. If you didn't do the conforming step, the subset, when you define the subset, that longitudinal data would be very sparse. So the conforming step really is critical linchpin to this being something that you can productionize and systematize across all studies. So again, you conform, you do that as part of study setup, and then the subsetting stars are automatically generated through an interactive experience. The end user can actually build out these subsets, and at that point, you can take your standard library of methods, or you can write new methods against these subsets. So you can take old methods, run them against subsets, new methods, run them against subsets. You can create new subsets and take those methods and have a record of all the different subsets that you've created. And actually, you can take the results of those methods, and those get captured by LSH as well, so that you can distribute results, which may be PDFs, tables, etc. So now I'm actually going to take a minute to talk about the technical framework, because there really wasn't a focus on products so far in this discussion. Okay, so let's see. I did just get a question here, and let me see. Just give me a second to read it. R and SAS scripts are stored in LSH. What is the format? How do you run your script from LSH instead of R or a SAS terminal? So one of the features of LSH is that you take the code from something like SAS or R, Informatica, or PL SQL, and you store the code under version control in the LSH framework. You then can take that what's called a program definition and attach it to a set of tables. So you're not copying the program just because you're attaching it to a set of tables or data sets, you're actually creating what's called an instance of that program. So the program sits in the library, your program code is there, and you're pointing it to a set of tables. You can then submit that program against those data tables, and LSH, if in these supported technologies, will in fact, in a very controlled way, in a secure way on a batch server, probably a Unix server depending on the technology, will actually execute that code against that set of data, and will take the results of those data sets or data frames in the case of R, upload the results back into LSH, clean itself up from a security standpoint so that you have the code, you have the record of the job, what data it ran against, as well as the results of that, whether the results are data sets or whether the results are PDF logs and list files. And they all come back into LSH. LSH itself doesn't do anything at runtime except for call a SAS engine or an R engine to execute the code. So in effect, when it's actually executing, the code itself is somewhat ignorant of that LSH even exists. Similarly, when you're developing the methods, you want to develop them interactively. You launch into a development session with your subject subset as your source. You're just in your SAS development environment coding away, hitting submit, or calling submit over and over and over again against that data. And really only when you're happy with that code would you load it back into the Life Sciences Hub. So it's sort of a technology framework to keep all this stuff under control and capture the results. But in fact, the native engine that you code under is still what executes when it executes in batch. So let's talk a little bit more. That leads nicely into what is the proposed environment, because it's not restricted to LSH. So this is a proposed environment for building out this kind of infrastructure. There are other possibilities, so this is just to give a flavor for it. So we're saying the overall framework for managing the data, handling all this conforming, auto-generating the subsets, putting the star schemas together, and saving the results of the analytical methods is the Life Sciences Data Hub. We really see R and SAS as the primary tools for analytical methods. It's not restricted to that by any means, but in all the practical discussions I've had with companies, those are the only two tools I've ever really seen anybody work with for actually building out these analytical methods. For that patient population subsetting experience, while the data may sit in stars inside LSH, LSH doesn't come with a way to look at that data in that interactive way. Really, Spotfire and OBIEE are probably the two best tools on the market, but certainly there are other tools out there for doing this ad hoc kind of exploratory analysis on these subsets, but those are typically the kind of tools. And those tools are essentially integrated with LSH. In terms of conforming data, or bringing it from your EDC source structures or your legacy source structures to the conformed structure, we've seen Informatica, we've seen SAS. There are mapper tools out there. SQL is another transformational tool for conforming the data. Typically, the group that conforms the data is not the group that has anything to do with the analytics. The people that conform to data tend to be a little bit more focused on the database build on the EDC side, and so they know about their study, and so whatever technology they're familiar with. Whereas obviously if you're building analytical methods, SQL is probably not the best one, and Informatica is probably not the best one. Technically you could do it, but that's not really where the skill set lies within the industry. Okay. So I'm not going to go through all the features of LSH that actually enable this, but I'm going to highlight a few of them. So for the rapid acquisition of data, so for pulling data from EDC systems, there's a lot of out-of-the-box functionality to just basically load data sets, load from Oracle Clinical, pull in data sets from Rave, Inform, etc., and actually get that stuff in there as is. Also, if you have CTMS systems, outcomes data, whether you can just bang the data into LSH pretty easy. But then it's still in the same shape that you got it. It may be into the controlled environment, but it's the same shape. There's lots of functionality around auditing and blinding and handling PK data and PD data. You can even create a federated view if the raw data that you're staging in is on another database. You can actually just create a pass-through. You don't even have to physically stage the data into LSH. So that takes care of getting the data in as is. The next step is to conform the data. So one of the things that's important is that the Life Sciences Hub supports multiple parallel data models. So I could have many SDTM type data models, study specific, Janus, any combination, company-specific, atom, they can coexist along with the data models that have to do with the star schemas and the data models that have to do with your analytical methods that you want to work with. So you're not restricted to a specific set of data models. There's a lot of governance in there that actually lets you control that with security, but you don't really have a restriction on the number and types of data models that you have. We already talked about open technology. I'm not going to go into that. There are version control, snapshots. So it's really an industrial strength application for dealing with companies rather than individuals. You wouldn't need to go through all of this trouble if you had one programmer with one set of data that they were working with, because they're not really focused on reproducing things over a period of time, of building up libraries of knowledge. One of the key factors and features of the Life Sciences Hub is there's a series of programmatic APIs that basically can build out programs and build out schemas algorithmically. And at BioPharm, we've taken advantage of that by being able to automatically do things like add studies to dimensional models. We can automatically generate the longitudinal data given on subject subsets because there's a design pattern to that. And you can write a program that says, hey, if you give me a set of subjects and a set of studies, we've written automation using these APIs that can just go into the work areas that contain all the conformed data, bring it all into one local area, and automatically pool that data for you. Now, you could do that by hand, but because there's a pattern to it, BioPharm has written some utility programs that actually bring that all together. So that was the primary area that I wanted to cover. I still have a few minutes left. Basically, just before we go into the Q&A, just need to throw up an advertising slide. So BioPharm does handle basically all the business case requirements, analysis, installation, configuration on Life Sciences Hub, hosting, a product called Clinical Development Analytics, which we're not talking about today. So we are involved in helping implement systems like this in environments like this. And I'll just put up the contact information on the screen at this point. And I would welcome, if anybody has any questions at all, to please go ahead and type them into their chat window now. I'll give people just a second. We don't really have any questions at this point. I would like to thank everybody for taking the time. We are just about wrapped up for time right now, and I don't have any active questions at this point. If you are interested in digging a little deeper and understanding any of these steps, whether it be just how do I bring data together and conform it, or how do I look at some very sophisticated, how do I get the full value set of managing this risk analysis and monitoring these subjects and for risk, doing this enrollment modeling, etc., we can certainly talk in detail. We certainly will make this presentation available. We will not be actually delivering the slides as standalone slides, but what we will be doing is each one of you that has joined here will be receiving within 24 hours a link that will let you replay this presentation. And in addition, if you have colleagues that want to see this presentation, they'll be able to register after the fact, and they will be able to replay this presentation with the audio as well as the slides at their leisure. But we don't actually distribute the slides like as a PowerPoint deck or something like that. With that, I think we're just about out of time here. Again, thank you very much for joining this call. I hope you found it informative, and please feel free to reach out to us and contact us, and we definitely can help you with working through these types of questions within your organization. So thank you again.